Okay, I think we are live now and we're alive, so that's wonderful too. So welcome everybody to the curatorial talk of Lyndsey Walsh regarding the exhibition of the Art and Natural Science Master Showcase 2021. My name is Laura Rodriguez, and I had the honor to be teacher assistant during the course of Lyndsey Walsh in the Master of ITMO University. And during this showcase, the students presented their projects where they have cared for, collaborated, and created with both humans and non-humans agents, living and non-living organic materials, and other natural process from the biological sciences. It's my honor to host this curatorial talk with Lyndsey Walsh that guide us during several months through the practices of bioart, genetic biology, and tissue culturing. Lyndsey Walsh is an American artist, a designer, writer, researcher, and cultural worker based in Berlin. Although she disagrees with the labels and extreme categorization, we could say that her works are primarily categorized by her creative practice in the realms of biological art and biodesign. She has an incredible career. She holds a bachelor in individualized studies from New York University and a master's degree in biological arts with a distinction from Symbiotica Center of Excellence in Biological Arts from the University of Western Australia. Her works have been exhibited internationally all around the world, and she has publications with the Nautilus magazine, Cloth magazine, the Goethe Institute, and more. Lyndsey is captivated and enthralled by the creatures emerging from spaces in between and crossing over the imaginary, the becoming, and reality. Her work explores the cultural and social quagmire of disease, identity, the body, human and non-human relationships, and speculative narratives on the future. She is a social media manager or creative producer from the Dutch nonprofit arts organization Unborn Collective. And regarding this issue, she also delivered for her students a workshop in how to write popular science. And she is half of the collaborative duo Crawlers, along with Australian artist and writing Jess Cockerill. Currently, Lyndsey is a visiting scholar and researcher within the Department of Experimental Biophysics at Humboldt University in Berlin. And she is also a professor at my university and part of the A residency. So without furthermore, I'm very happy to be here with Lyndsey to talk about this journey that together with the students we passed during the last years and now is culminating in this beautiful showcase presentation. So Lyndsey, the stage is yours.
Thank you so much, Laura. And yeah, that was a wonderful introduction. And Laura has been a big contributor to this course, not only as the teaching assistant and as my co-curator, but also as one of the artists in the exhibition itself. So I'm very excited and thankful for her presence in my life.
Thank you.
Yeah, so I feel as if the works in this exhibition are particularly special because there was no sort of exhibition open call to gather these works. These works were really, the guidance that I had and the involvement between me and the students was start from finish about formulating ideas to their production and now to their final exhibition, which is really something special, I think, to take part in as a curator and as a professor and also as an artist to see kind of emerge. And so for some of the artists in this exhibition, it was their first time going into the lab and getting their hands wet and dirty with living materials and scientific practices, as well as for some of the other students in this course, it was their first time making an artwork ever, which is really exciting to watch someone do for the very first time. So kind of to introduce the inspiration for these works, I thought it would be best to sort of kind of give this overview of what the Art and Natural Science course was. And so this was a practical course taught last semester at ITMA in the Art and Science Center for second-year master's students. The course itself was really aimed at giving students both this practical experience with sort of the skill building in the laboratory, while also incorporating different art and science-based themes and topics. And having discussions around those. Focused on the rise of bioart and biodesign, as well as these other sort of transdisciplinary creative ventures, and the following artistic approaches and techniques that have emerged from these practices. And because this course took a practical approach, really the students were exploring the ways in which artists care and collaborate and create with both human and non-humans, as well as the different living and non-living organic materials you can use in a creative practice, and other types of natural processes that are very common for study in the sciences. Some of the required readings in this course were works by Oran Katz and Yonat Zur, Tosh Bates, Sean Ross, Evan Kurtzky, Alexandra Daisy Ginsberg, myself, as well as Rizard Klusinski. So in light of Corona, we began this course kind of looking at how artists and designers around the world are adapting their practices to working at home, which is thought to be almost impossible when you are working with living materials, especially things that are quite hazardous. How do you bring your lab to your house? And I'm sure many scientists as well have been struggling with this issue about what to do with their research if they can't go to the laboratory. So kind of as a response to this, to an article published with Umbor Collective by different students from the University College of the Arts in London's Material Futures program, they sort of, the students in this article were kind of documenting how their practices were changing now that they were at home. And we then in class had a nice discussion about what the students can do in their own homes, and if they even had materials lying around in their own homes, which they could use potentially following different DIY bio protocols, which was really interesting, kind of like led them on a very bizarre scavenger hunt, I guess, around their kitchens and things like that. Our next lesson that we really covered was talking about aesthetics of care. So when working with living materials, artists are required to a certain extent to care for them. Sometimes the point of the artwork, I guess, is to not care for them, but in this instance, we're focusing on the caring aspects. This, of course, transitions then into the aesthetics that the artist uses in the final production of the artwork. And so artists and designers typically make aesthetic decisions about whether or not to reveal these systems of care in their final piece. And in their practices. And so we were really looking at this reading of Aesthetics of Reason and Care by Rizard Klusinski, where we explored how different artists and designers approach notions surrounding care in their own practices and artworks. So in the picture here, we have the piece Victimless Leather by the Tissue Culture and Art Project, which is made up of Yonat Zur and Oran Katz. And so in this work, Katz and Zur make the systems of care that maintains their victimless leather piece clearly visible for their viewer and their audience. And they also utilize sort of scientific aesthetics to support the piece and the overall conceptual meaning about the speculative vision they're creating in a world where victimless leather is grown in a laboratory. The kind of key to this aesthetic development in this artwork is revealing the feeding mechanisms for the piece. Katz and Zur use fetal calf serum, which is kind of central to their artistic critique in not only victimless leather, but also in their other works such as Semi-Living Steak and Disembodied Cuisine, and how fetal calf serum may not necessarily be victimless at all. And so by highlighting sort of this material needed support for the care for their piece, that's really where their critique kind of lies in the final production of the artwork. And this is something that I, as an artist and designer myself, have my own experience with, which I shared with the students. In my work, The Hanging Drop, I created performative sculptures using mesenchymal stem cells in the laboratory, this, which is one of the images to the left of your screen. This was done using human mesenchymal stem cells, which are stem cells taken from the stroma of the bone marrow. And in the gallery, I ended up using, due to ethics and biosafety regulations in Australia, I ended up using mesenchymal stem cells from rats. And as an artist, I chose to reveal to my audience kind of the depth of my materials, as well as aesthetically position the work that really focused in on this sort of trapped nature of these performing sculptures that were in the incubator in the gallery. Unlike Katz and Zur, my work does not show the feeding mechanisms taking place to care for the cells, which really was done to support my overall artistic critique about the sense of doom lurking behind these visually beautiful sculptures that were being created in the gallery, but then were forced to eventually die as they couldn't be taken out of their sort of suspended droplets. And then this sort of brought a discussion into on a more organismal level as well, where I introduced the concept of a model organism to the class. Model organisms are organisms that are used in science for research and often stand in for the human body. Therefore, the caring system surrounding these organisms is really centered on their role in scientific research to sort of serve human desires and human directed outcomes. One of the major artworks of our discussion focused on Tasha Bates' work in vitro, which positions caring systems of model organisms by bringing them into the gallery space so viewers can come and see the daily caring tasks that are being performed by Tash. And while the organisms in this piece serve as performers for human desires of an artwork, the focal point of Bates's labor really to care for the organism sets out to challenge this typical role that these organisms play in science. And so following kind of our dialogues about aesthetics of care, the next themes of the course shifted more into this discussion about ethics and biosafety. This was both a necessary sort of induction to the upcoming work that we were doing in the lab, as well as a prompt for the students to begin thinking about the ethical decision-making in their own creative practices. And by working with living materials, artists and designers are often implicated in a paradox of sorts about how you can kind of reconcile with your own creative practices sort of use of living organisms and living materials, and also the sort of exploitative effects of this. This is something that I think any bio artist or artist working with the living materials is often asked about in any artist talk or Q&A as well. So when engaging in this discussion, one of the questions that I ask the students is if aesthetic outcomes could be considered ethical. And this question can kind of be interpreted in a number of ways, which it did in our class as well. And one being, do aesthetic outcomes justify the means needed in order to produce a work? Or can an artist apply their aesthetic decision-making to also be transparent of this ethical decision-making in their practice? As we don't live in a vacuum, and we are also in an age of things like cancel culture and sort of these global calls of greater social reform that are really important, it's really a good reminder about the implications of our individual actions. And as artists, when working with living materials, to kind of, you need to answer these sort of ethical questions that your work is creating in some sort of way, either through your aesthetic choices and decision making, or through your artistic critique itself as well. And it was also important for the students to consider how they could take potential responsibility for the actions that they were engaging in. So for example, in the slide here, we have Joss Ehner, who's kind of notorious for creating these DIY CRISPR kits, but has also gone on record for regretting his sort of aesthetic decisions to publicly inject himself with CRISPR. However, yet his DIY CRISPR kits that he's made are still for sale online, which leads to a number of other ethical questions about kind of how sincere this regret was, and is this more of a publicity stunt with all these articles and coming out and renouncing this, and kind of like leads to certain, I don't know, ethical questions about really how does Zainer take responsibility for his actions by making these kits? And this also brought up a really important question surrounding safety, which I take very seriously, especially as an artist who works in the lab. I get asked about safety all the time in my work by both the general public and by scientists. And yeah, there's the very obvious reason for an artist to take safety very seriously, which is the fact that other scientists will not work with you if you don't follow biosafety guidelines that are required in their lab. But also, I think it's really important to consider safety and different hazards in any practice where you're working with physical materials in the physical world to an extent, and also safety in terms of data privacy if you're then working in the digital world. So for many of the students who did come from a more digital or curatorial background, this was tricky to think about. And outside of my scientific background, I was very lucky to have worked professionally in the film industry and with heavy machinery for construction. So it was really second nature more for me to think of identifying different hazards, like don't put your fingers under a bandsaw, and like the chemicals that you use need to go in a chemical closet because they're flammable and things like that. And where's like the fire extinguisher? But for a lot of students, this was new to like walk into a room and identify hazards and things that were hazardous to them. So a big point of the discussion that came up in the course, which you'll see later on why in their artworks, was that bacteria are hazards. And even when they're sampled from your own body, once they reach that petri dish, they must be treated as dangerous substances. And also in the age of Corona, even your samples, once they go on a Q-tip or a swab, that is now a hazard because it contains other sort of things that could be hazardous to other people. And appropriate protective gear and disposal methods must be applied. So as a result, the students all had to submit ethics and biosafety applications in order to exhibit their works in the gallery. Which also outlines sort of plans for handling consent and privacy and waste disposal of their pieces. And they also had to plan how they would create different levels of containment within their artworks that featured different biohazards. So these are either done through physical barriers of having double containment, which I think if you've been to the exhibition, you'll see we have like lots of sort of acrylic and glass structures surrounding a lot of the works. Or it was done with sort of psychological barriers where a person may walk into the gallery and sort of there's this certain barriers have been set up psychologically for people to not touch things, either by having gallery attendants or sort of indicators about where you can stand, how far away from a piece. And so our next kind of thematic discussion that we got into in the course was concerning notions of biodesign. Referring to the wonderful book edited by Alexandra Daisy Ginsburg called Synthetic Aesthetics, I challenged the students to think about how scientists and designers are actively reshaping what life is through genetic modification techniques and creating novel organisms, such as these semi-living entities that have been kind of characterized by Katzenzer. In Ginsburg's book, she often refers to like dissecting these notions of better. And so this was a really interesting conversation to bring up about like, is there one better form of life? And should we design for better when in reality, organisms exist for many, many, many different reasons? And comparing the two, what will the world look like if we keep designing organisms for sort of like human needs and desires versus other needs and desires? And should we do this to begin with at all? So accompanying sort of this thematic discussion, the students ended up exploring different biological-based materials by learning how to grow and manipulate kombucha leather, and also took how to create different sort of constructs of calcium alginate mixture. This material exploration was done to provide a foundation for incorporating biological-based materials into their creative practice, and directed to give students kind of a lot of a freedom to push what they could do materially with these substances. As you can see here, this is a piece from Laura. She ended up knitting the calcium alginate structures, which was very cool. And so they could play with different types of rigidity and different textures they could do with all these different types of materials. The students also ended up incorporating biological-based material approaches in their lessons on tissue culture techniques. In the laboratory, they decellularized apples and spinach leaves, also preparing some sort of calcium carbonate structures taken from the cuttle bone that's inside of a cuttlefish. And they later ended up seeding these with C2, C12 cells that were derived from a mouse. One of the big lessons that the students ended up learning during these tissue culture experiments was like how easy it was to have your tissue culture work contaminated, and how needy tissues are in the lab and how much care they really do require in order not to die. So this really led to some wonderful discussions about how to work with such fragile materials and kind of how to design your creative practice to really deal with these sort of inherent disappointments that exist and failures and senses of uncertainty about sort of the material potential of your practice. We then switched gears to working with organisms like the potential source of our tissue culture contamination, and kind of began some material exploration with microbes. A large point of our thematic discussion centered on the role of queer theory in discussing these sort of multi-species entanglements and deconstructing our notion of what the human body is. And we also talked about many different desires, including different sexual desires that exist within the human body of the other organisms that live there and contribute to the overall form and function of the body. The students then had a chance to explore the different materials they could use to culture bacteria. These were done using a variety of different types of agar plates. They sampled different bacterial sources and grew their cultures in the lab, testing out how their bacteria would react to these different types of agar and sort of what, here a lot of the color production that has been done is based on the sugar consumption of the bacteria, with particular indicators in the agar. And so they were playing around with like where they could sample bacteria from and how that would create particular kinds of reactions with different types of agars, which was really interesting. And definitely, I think a lot of students enjoyed this most with the course and kind of incorporated this into their final artworks. The last theme brought up in the course was concerning non-visual modalities in art. And while I think that we all tend to rely on sort of the visual nature and themes of art, especially in the fact that a lot of our arts is called under this umbrella term of visual arts, there are many artworks that are actively deconstructing sort of this reliance on the visual senses. For example, this piece here is by Gail Knowles called World Sensorium, and it is a smell-based artwork that creates a signature scent of every country's representative smell, which has been blended in accordance to the population size of that country. This work was a UNESCO endorsed heritage piece as well, and during Times Square in the year 2000 when the ball dropped. So while talking about these non-visual modalities in art, this sort of led us into a wonderful discussion about disability studies and how we perceive and assume particular things about our senses and other people's senses in society, and how experience can really go beyond just our visual sense and how different people experience things in different ways. So for example, one of the artworks we talked about was Loud Cane 1.0 by Carmen Papalia. And the work creates a walking cane for the visually impaired that translates information related to texture on the ground into sound, which is really quite an interesting way to really deconstruct this visual reliance. For our workshop component, we focused on the non-visual sense of smell. Together, both Laura, I, and the students, we all engaged in this sort of sweat-producing activities, and then extracted odors from these sweat samples using steam distillation to create body odor perfumes. This was an especially interesting workshop to do in Corona times, as we kind of had to take into consideration the necessary heat we needed to get in our steam distillation process in order to kill the coronavirus, if any existed. And yeah, the students especially ended up creating a bunch of corona safe and free body odor perfumes, which was wonderful. So now kind of transitioning from our more course and practical based works that the students did, I would like to introduce sort of the first artwork of the exhibition, which is the piece titled Golem by Sofia Ospanova and Anna Kaplan. Sophia and Anna decided to work together very early on in the course, and they were really inspired by the figure of the golem as this sort of narcissistic myth invented by humans, which was kind of driven by this desire to become a master of matter, and for the humans to be able to revive matter, subdue it, and control it. And so as artists, they kind of sought to reanimate a clay figure by repositioning the bacteria living on the clay particles as living matter and making them into these active agents of this sort of living corporality of their art object. The projections seen over this clay figure are taken from the imaging of the bacteria cultures inhabiting the clay as well, to help sort of transition us from this macro and micro lens as the audience. Moving on, we have Dina Zatova's BioArch. The aim of BioArch project was to kind of search for new forms of architecture that were inspired from both this microscopic landscape that Dina was researching. And Dina sort of conducted a lot of material research using bacteria and fungi that she collected from buildings and surfaces and people, and created different calcium alginate structures representing the material elements of this microscopic world in our gallery, which was really cool. The next work we have is Victoria Gopka's Tower of Babel. The Tower of Babel project that Victoria made was really her reflection on sort of the post-humanist body, and trying to also critically re-examine this, this biblical Tower of Babel narrative. Victoria ended up taking different bacterial samples from her body and embedded these sort of samples into a agar structure that made up this tower. She really sought out to reflect on notions of incompleteness and this constant flux of bodily changes in the tower's own construction as it shifts over time in response to the growth and proliferation of the different microorganisms inhabiting it. Then we have Natalia Molina's Skinless Mouse. So this sort of sculpture was really looking at both the history of the C2, C12 lines, and also sort of the ethics of working with non-human model organisms in the lab. By using a plant tissue matrix, which is positioned to be right before it's seeded with a line of mouse myoblast cells. She really sets out to question sort of how we work with these new organisms when they really are defenseless in a certain sense, especially in the tissue culture realm. The skinless aspect of it, the mouse in the lab in this sense, so abstracted from its actual biological form, has no defenses at all to take care of itself and is completely reliant on this techno-scientific body and human care and human actions. And then we have a work from my co-curator and teaching assistant, Laura, Becoming Plant. And Laura was really inspired by the decellularization processes we explored in the course. And so Becoming Plant kind of sets out to sort of envision this speculative narrative of body modifications that occur, where people can kind of take on sort of the forms and structures of plants into their own bodies, and really sets out to expose sort of this transparency and fragility of the shifting nature of body image and form. Laura was also really inspired by Guattari and Deleuze's kind of Becoming Animal as well, and the philosophical teachings surrounding that. And yeah, I'm sure Laura also, Laura, it's hard to talk about your work when you're right here. I'm sure you have way more to say about it than I could.
No, thank you very much.
It's really wonderful. I'm obsessed with Laura's use of gore here. It's really totally in my horror aesthetics as well. So this is a really lovely work to see be developed and come to life.
Thank you for the opportunity.
Yeah. And finally, we kind of have a group piece made by all of the artists, which was also being assisted by our scientific collaborator, Anastasia Kuchakova, titled Living Labor. So this work, the students kind of did another workshop outside of my course where they were harvesting spider silk. And they really set out to question how we consider the labor of non-human agents through sort of this anthropocentric lens. And so the spider silk, which is secreted from the spiders that they were harvesting from, is very necessary for the spider's own labor that it needs to capture prey and create cocoons and protect their interspecific communication channels. So the web is sort of this both very like a tool for the spider as well as a prosthesis that it expands upon their sensorial perceptions. And so they then ended up taking the silk and making it magnetic by incorporating these sort of ferromagnetic nanoparticles into it. And together they ended up creating this very uncanny and uncomfortable sculpture that moves. It's very creepy in a very fun way. And kind of takes on a life of its own, sort of embodying the sense of uncanniness about this displaced labor that now exists from the silk that's been harvested by the spiders. Yeah. So those are our works in the exhibition. Both me and Laura are very proud of the students. We really are so excited by what they came up with, especially since we gave them very minimal kind of prompts and guidelines to like produce the work. That we really wanted them to take whatever they thoughts they had and ideas they had and sort of run with it and play as much as possible and experiment. I think that was some of the biggest things we emphasized, was to really, especially since it was their first time creating works of this nature, to like really get their hands dirty and try out as many things as they possibly could to then form this final artwork. So we're both very proud and very excited that it turned out this way. It's very wonderful and beautiful.
Yes, I totally agree. To see all the development from the concept to the prototypes and then to the final showcase has been very excited. I'm also very proud of all the students. I think it was a very complete course because it not only prepares artists, but also if our students planning to go to the curatorial studies, it's very important to understand how to work with bioart, or even if they want to be the managers of galleries, they definitely got an insight of how to work with living matter and how to exhibit it. And so I think the course itself was wonderful. Thank you, Lyndsey. And then that was reflected in the showcase that we have now. So we are open now for questions. You are free to write in the chat in the YouTube channel. And if there is any question, I will read it aloud and we will try to answer to all of them. Meanwhile, I would like just to make some comments about the final pieces. For example, when Lyndsey was talking about this sphere with the magnetic silk, and we put it in the image. Yes, it has been very interesting that because it moves, given that there is magnets inside rotating. Some people have believed in the beginning that there is spiders inside or some insects. It has been interesting to see the reaction, and I think that, as she said, is quite uncanny, but it fits with all the exhibition. We really have a non-human exhibition, and that has been very interesting. When we see Victoria's Tower, it changed from one day to another. So if you visited the gallery, maybe in the inauguration, now you can see a totally different structure because it's a living sculpture that is changing. Now we have some bubbles because the bacteria is growing. And for me, one of the strongest measures from the course was the ethical approach the living organisms, and I think most of the students consider it in their artworks. And as example, we have a skinless mouse from Natalia that went deeper to the history and the ethical issues of working with these cells, because the mice that humans took the cells from, he died decades ago, but we still have the cells. This is a discourse that we analyze in the human context with Lyndsey when she explained about HeLa cells that are human cells that also are now all over the world. And the fact that the student translated into the animal landscape is also very interesting. And then Dina has a architectural background. So for me, it was very exciting to see how she make the link between her career as an architect and now as an artist through biodesign. So I think that was really cool. And also for me during this course, maybe because we started with a do-it-yourself lesson or because we were in pandemia, but it really demystified the idea that to make bioart you should have a lot of equipment, expensive laboratories, and so on. Of course, it is really good to have all the equipment. If you can work in a lab, that facilitates a lot. But my project born in the kitchen. So
that's right, you made it entirely at home pretty much.
Yes.
You were the true lockdown lab.
And we found this do-it-yourself protocols. Lyndsey explained with it could be done to the cellularization with DNS, that is a very strong chemical compound, like very strong soap. But she also explained like it could some protocol from peeling, if I remember correctly.
Yeah, Andrew Pelling. He has some wonderful published protocols online. If anyone is interested in trying to get involved in their own makeshift DIY biolab, Andrew Pelling's always my go-to for protocols.
So yeah, I started with this do-it-yourself protocol, and then in some moment I have all these plants, all these leaves in my kitchen window. And when we have a small dinner with a friend, so he came to the kitchen and was like, "What is this?" And my husband was like, "Yeah, Laura's project." So it really demystified this and opened up a lot of opportunities to work as an artist, even from home. So that's what I thought. So we have a question. Regine Rapp, we would like to know more about becoming plant. Okay, then if it's okay, I will try to answer this.
Go for it. It's your work. Take it away.
Thank you. So with this, the cellularization process, we learn how to remove the cells from vegetal tissues. So and then we have, when we remove the cells, we have this transparent structure made of cellulose. And in the laboratories, there is experiments when they are trying to put new cells over these scaffolds, and these new cells could be from other species like mice or human. So that opened up the opportunity for a speculation that when we could use these structures as implants for the human skin. So personally, I took the idea of the cellularization as a deconstruction, and the question of what is the essence of the plant if we remove all the cells, and what is the essence also of human. So when we also separate each other as by the forms. So when you still see this transparent leaf or flower, you kind of associate that it's a flower or a plant, but there is no cells anymore of a plant, of a flower or the plant. So what happened if we acquire a different shape that we can talk about the malleability of the body, the the incompleteness or ability to change of the forms, and how to deconstruct also these ideas of how something should look like to be something. So then also the idea of becoming with other species, that is when we interact, when we are like in a unity with another species. So if we are kind of absorbing the form, what does that mean for us to become with this plant? So the project become not only speculation of implant, but as a ritual to how to become with a plant. So there is instructions after the surgery that a person should follow, because now it's not only a single entity, but now it's becoming with another species. So that become a little bit funny to think in what it means to become with a plant, and not only like imitating behavior, but also psychologically what it means to be with other species. I hope I answered.
Yeah. No, I think also you really embodied the sense of play with the piece as well. You can definitely see like Laura tried so many different things in this whole sort of technique and process making. She tried flowers, she tried leaves, she tried different kinds of plants. And like you could really see how the work kind of like brings all of your sense of like experimental material play into one cohesive narrative, which is really nice as well. From the side who saw the whole process.
Thank you. Okay, we have another question. Dear Lyndsey, thank you for your wonderful talk. Can you tell a little bit more about art and science perspective in disability studies?
Yeah, for sure. So I mean, once again, disability studies it's really hard to kind of encompass all of it as well, because like everyone's perspective is so limited. I identify as a disabled artist, but like once again, my disability is very unique to me. So the way that I like perceive and interpret the world around me is very much contained to my experience of it, which I think sometimes is why we have difficulties reconciling with inherent ableism that we have in certain practices and the ways we interact with people. And it's very, very difficult to overcome those sorts of things and kind of like go outside of ourselves to really like seek out to accommodate different experiences and abilities. But in science, I think it's really interesting for me personally as a disabled artist to sort of kind of set out to more take back narratives in science and in medical science, especially when the bodies of disabled people are some of the most heavily sort of oppressed and exploited by those systems as well. There's also some wonderful works by friends of mine who do this. There's a piece called Cryptologies by Romley Alice Walden, which is a wonderful piece about how Romley Alice experiences the world from her while not being able to go outside very often or go outside very far and things like that. So what does ecology mean when you're oftentimes homeward confined and bound, and how do you then experience the world and define ecology when you can be quite limited in your experience of it? It's a really beautiful piece as well. Yeah, and so I think having disabled artists come in and take sort of this narrative role on science and in medical science is something really beautiful, and I hope more artists do to represent that as well. I think it's also very tricky to then, for other artists who maybe are not from that community, to kind of narrate those experiences because they don't have that perspective. But I kind of hope more from a curatorial standpoint that there will be more sort of accommodations for and open calls available for disabled artists in the community, especially in the science, art, and biological art community, to come in and really take back narratives in medical science about bodies and body image and things like that. I hope I answered the question. That was kind of me waffling about various directions. but
I think there is a very interesting and complex topic. That's why there is a lot to talk about it.
Yeah, it's something that I also personally struggle with, because like I have a piece that talks about like this impending disease that I may have in the future that I will be working on with ITMO in the fall, but I don't have any pieces that talk about like my actual disability that I currently experience. So it would be interesting. I don't know if I'll ever like reveal that story just yet, because it's something so personal and it's always like very difficult to talk about, and it's as well. So there's like this duality of like what to share, how to share, because once, especially with invisible illnesses and invisible disabilities, you always get people kind of questioning how the legitimacy of it. Disability isn't just an inability to function like and to perform actions like other people, but also how people perceive you as being able to do that. It kind of has this dual role to it, which is quite fascinating to be in the middle of, but also outside of as well. So yeah, that would be my more personal take, I guess.
Thank you very much. Yeah, meanwhile you are talking also, you may maybe think in the beginning, or like since 1 year ago, I have been also thinking about different behavioral patterns of like what we call like mental illness, how like would love to explore this through art and with a scientific background. So I think society needs this type of art, and I'm happy that you are addressing it.
Yeah.
What is it? How it differs the experience of creating your own artworks and guiding students to create their own artworks?
Oh, that's a good question. Yeah. There's definitely like the biggest difference, I guess, is like the unexpectedness of working with students and creating artworks, because like although I often get into projects, I have no idea what the outcome will be at all, just because maybe I don't plan enough. But I think my living materials tend to like run away with whatever my idea is as well. But students, on the other hand, they really like, they're so, they have their own ideas, which is great. All students should do that. This is how you become an artist, is you get very stubborn about your ideas and you go out and you do it. But it's always like so surprising to see what they end up coming up with, and what they found to be important to them, and what sort of ideas really resonate with who they are and what they want to do. That's something that I always find to be quite fascinating. Whereas like when you're doing it yourself, you know yourself very well, I hope, for the most part. Sometimes you surprise yourself, but like, I don't know. When I make artworks, it's all things that I've obsessed over like time and time again. So I find them to be very mundane and tiresome by the end of the piece. Whereas like when working with students, I'm like, oh, I never thought about that way. That's really interesting to consider, which is nice.
Great. Okay, so let's wrap up our curatorial talk. Do you have any more comment to not only the students, but the people interested in bioart here in our community?
Yeah, for sure. I think like getting involved with the Art and Science Center at ITMO is a great way to get started if you are really interested in these sorts of things and topics as well. I think this course really shows that like anyone can get involved with these ideas and concepts regardless of your background. The number of times the students were like, I'm not a scientist, Lyndsey, I'm not a biologist, like, and that sense of uncomfortableness was expressed, but like I think that's something that can definitely be overcome, and you can learn how to like interact with these sorts of materials and ideas as well. You just have to have the passion and like the drive to kind of do it. And so I'd like to extend that sort of sentiment, I guess, to the greater community as well. Yeah.
Thank you. Thank you very much, Lyndsey. And thank you for all the spectators in YouTube. There are some questions. They ask us if we can put the names of some of the artists you were mentioning in the disability studies. So yeah, I will add after the call into the YouTube comments some of the artists that we mentioned. And thank you very much, everyone, for your time. Thank you, Lyndsey, again for all these months and for this awesome hour.
Thank you. Thank you for having me. And once again, I'm so proud of all the students and their work, and I'm honored to be able to present this talk in the first place for them. So.
Thank you. So we wait for you, all of you in the gallery until 14th of February. You can check out the information of the exhibition, the showcase and activities we have in the Instagram Air ITMO and the Art and Science Center. Thank you very much. Bye-bye.